I must say it's a really pleasure to be here and have the opportunity to talk to you about my previous research that I've been doing for the past 10 years or so. And it is something it's here, sure. And what really got me interested in studying puberty and the beginning of the reproduction in heifers is that when you look at the amount of nutrients you use to produce a kilogram of beef, you can get up to 70% of the total resources being used just to maintain the cow herd, right? So that's highly inefficient. So we need to do something to change that, and those numbers are from Brazil, of course. And the reason for this high inefficient use of resources is that you have low fertility of cows, around 60% is the average for Brazil, and a high age at first calving, right? Usually above 36 months. And as you can see in this table, if you have a longer age or higher age at first calving, you need to maintain much more heifers just to replace the cows in your herd. So if you reduce from 36 to 24 months, you can have a drastic reduction in the amount of heifers you need to maintain in your herd, right? So not only that, but when you are working with Bosindicus breeds, they are usually more late maturing than Bos Taurus breeds. You know, this is a work done here in Australia by Rodrigues and others, comparing Brahma cattle with, I believe it was shorthorn cattle. And they were raised on the same conditions, the same nutrition, but the Bosindicus heifers, they reach puberty much later and with a greater body weight than the Bos Taurus animals, right? So for the outline of the talk today, I'm going to tell you about the effects of nutrition, body fatness, and beginning of reproduction. The hypothesis that leptin is a signal for body fatness, and I will describe 4 experiments that we've conducted trying to answer some of these important questions. For example, why some heifers attain puberty sooner than others in the same condition? How can nutrition, high energy intake, hasten puberty in these animals? Can we induce puberty with leptin infusion? And we know progesterone is another way. If you add use a progesterone device in a heifer, you can induce early puberty. So what is the molecular mechanism for progesterone action on the hypothalamus, on the brain? So, and then we'll finish with some conclusions that take home points. All right, so we know that body weight is important. I just showed you a study done here in Australia. When you go to a farm and you set your target here, like 300 kilos is the body weight that my heifers will reach puberty. That's common for the Nylor breed. So then I develop my nutrition program around that so that I have a high percentage of heifers capable of becoming pregnant during the mating season, right? And body weight and body fatness is also really important. If you have thin animals, the success rate, expected success rate of these animals will be really low, right? So we know that. What amazes me about science is that for such an important and basic physiological pathway as the beginning of reproduction, right? We know that nutrition is important. We know that body fatness is important. And this is a quote from the Foster and Hillman at Nobel and News Physiology of Reproduction in 2015. We remain fundamentally ignorant of how growth is perceived by the central nervous system in many vertebrate species. It's not that we don't have any clue. We have dozens of clues how this works, but none of them actually holds. So this is really interesting. But not only that, after 1994, when leptin was discovered And then it became a possible link between nutrition and the central nervous system. Because when you remove nutrition, the animals are fasting for 2 days. There's no LH secretion in the pituitary, right? And if you infuse leptin in these animals, then you restore normal cycling, GnRH and LH secretion in the pituitary. So there is a possible link between body fatness and nutrition and reproduction. So then that led to the development of this hypothetical graphic model that with a greater caloric intake, you have higher body fatness and more leptin, and leptin would signal to the brain your amount of body fatness and then somehow initiate reproduction. And there are several examples of how body fatness can control reproduction, right? One example would be, we're just talking here before about high-performance athletes, right? So for marathon, female marathon runners, they usually have reproduction problems because there is not enough body fatness, right? So we know there is a link between body fatness and reproduction. We just don't know how. And putting in economical context in Brazil, there has been an explosion of artificial insemination in the past decades. So we are already giving a lot of protocols, hormonal protocols to these heifers. We're spending money, labor. So if we had some solution, some strategy to increase the fertility of these heifers or the cows, being with breeding or short-term nutrition strategies or a pharmacological solution to add to these protocols, I'm sure this would find its application in the field. So the first experiment is the one where we're looking at the genotype effects. Why some animals get pregnant and others don't. So when you look at the data from Rodrigues again, okay, there was a difference in the mean, but look at the range of body weight for these animals. You have some heifers reaching puberty with 280 kilos and others with 350, right? That's 70 kilos difference. That's 1 year worth of growth in the farm, right? The same for the Bostaurus breeds. So what is happening with these animals? That was our first experiment. And our hypothesis was that the late maturing Bos indicus heifers have less leptin expression in adipose tissue and or less sensitivity to leptin signaling in the hypothalamus. And this work was done by a master's student, Aline Vaiciunas. She was my first graduate student, and she's now on her second PhD at Tennessee. She probably liked too much the easy life of the grad students. So she was the one who conducted this research. We went to the experimental farm. We select 500 heifers, 100 of them born on the same month with very similar body weight. From these 100 heifers, we palpated them, select 15 heifers that look like they were post-puberty, and 15 that look like they were before puberty, so they have no corpus luteum. We injected them with prostaglandin to induce luteolysis and ovulation if they were pubertal. And then we select 6 that were clearly pubertal and 6 that were clearly non-pubertal, right? Very similar, same body weight, same age. Some were pubertal and some were not pubertal. And then we slaughter the animals and collect the hypothalamus and the adipose tissue to do real-time PCR analysis. We had a housekeeping gene, a control gene, ribosomal protein L19, and then we had leptin and the active form of the leptin receptor. SOX3 is a suppressor of cytokine signaling, and leptin is a cytokine. So SOX3 it blocks the effect of leptin on the brain. So our thinking was, if these late maturing heifers, they have a greater expression of SOX3, that would explain why they have delayed puberty. NPY it has an antagonist effect as leptin. So if leptin induces puberty, NPY blocks puberty. If leptin reduces intake, NPY stimulates intakes, right? So if leptin is Luke Skywalker, NPY will be Darth Vader. And then you have the receptors for NPY, right? So NPY doesn't do anything without the receptor. This is neuropeptide Y, so it's in the hypothalamus. And then you have the receptors. I don't know what the receptor for Darth Vader would be. Maybe stormtroopers or something, I don't know. So the first thing we saw was that the heifers that were early maturing heifers, they had much greater leptin expression in the adipose tissue. So they were producing more leptin per gram of adipose tissue, right? And that was in the omental and subcutaneous fat and not in the perirenal fat. That's good. And looking at the hypothalamus and the sensitivity of the hypothalamus to the leptin effect, there was no change in the leptin receptor. There was no change in the suppressor of cytokine signaling. There was no change in NPY expression, but there was a decrease in the expression of the NPY receptors. So the brain was less sensitive to NPY, right? To the bad guy, to Darth Vader. There were no stormtroopers in the brain. So the conclusion for this first experiment is that this experiment actually supported the hypothetical model that leptin is important because the early maturing heifers had more leptin expression, and also that the sensitivity of the brain to NPY was lower. And what we need to realize is that the brain is ready to reproduction since little after the birth of the animal, right? There is a LH secretion during birth and then a few weeks after birth. And if you inject GnRH to the pituitary, it starts secreting LH. Everything is ready to work since early age, but it's kept in place because of negative signals that happen in the brain. So if you remove the sensitivity of the brain to these negative signals, then it might induce puberty. Okay, so the second experiment was about nutrition. We know that if we feed the heifers with a high energy diet, they will reach puberty earlier. So we had two diets, one with formulated so the heifers would gain 0.4 kilos a day, low energy diet, and a high energy diet that would make the heifers gain 1.2 kilograms a day. 24 heifers, 19 months of age, 275 kilograms of body weight. Two master students, Marina and Giuliani. Both of them stayed for the PhD. Marina is now working at the University of São Paulo, and Giuliani went to work for a food company, and for the past years she was financing my research, actually. And the hypothesis was that high energy intake would hasten puberty through the leptin-NPY axis on the hypothalamus. We define puberty as the age, as the day that we first detect the CL on these animals by ultrasound, and this was confirmed by serum progesterone concentration above 1 nanogram per mil. So that was our definition of puberty, right? 8 heifers from each group were slaughtered on the second day after the CL detection. We collect the adipose tissue and collect the whole hypothalamus for gene expression analysis. This graph here shows the diameter for the dominant follicle. So both groups start with small follicles, about 7 millimeters. And as soon as they start eating a high energy diet, the follicles start to become bigger, right? So reaching the size of ovulation much faster. So this is the line for the high energy group, and this is the line for the low energy group. These animals start with the same age, the same body weight, the same body condition score, a measurement of body fatness, and then different average daily gain. So these animals' high energy diet ovulated at an earlier age than the low energy diet, with a similar body weight but greater body fatness. So the body condition score was higher for the animals on the high energy diet. And we actually analyzed the carcass composition of these heifers, and we know that there was a greater fat content on the carcass. All right. And also, leptin expression, so more fat and also more leptin being produced by the fat tissue, by all the three fat tissues. There was no treatment by tissue interaction in this case. They all produce more leptin with high energy intake. Right? So that's good. That is in accordance with our model. Looking at the hypothalamus, there was no effect of energy on NPY or NPY4 receptor or SOX3. There was a reduction in the expression of this RRB1 gene and NPY1 receptor, right? This ARRB1 gene is a beta-arrestin 1. It is involved in the regulation of G-protein couple receptor signaling, just like NPY and other bunch of receptors in the brain, right? So this protein here, it causes the internalization and recycling of the receptor, for the estrogen receptor, for example. So it's involved in signaling in the brain. And the NPY receptor, well, you know how that works, right? So you are, again, reducing the sensitivity of the brain to the negative stimulus. That's what high-energy diet is doing. And it's a similar effect as we've seen for the genotype study. So that's really interesting. So this experiment 2 also supports the hypothesis, more energy, more leptin, less sensitivity to the bad guys on the brain. All right, so now let's actually test the hypothesis and add leptin to the animals and try to induce puberty with these animals. So I was able to acquire more money and buy more hormones and do more research. Marina stayed for the PhD and did this project. So it's actually the same experiment. We had a third treatment. We are trying to trick the brain into thinking the animals were fat. So we had the low energy diet, the same treatment, but then applying leptin to these animals. Leptin was applied twice a day, subcutaneously, at a dose of 4.8 micrograms per kilogram of body weight. And this dose came from previous experiments showing the diurnal cycle of leptin in the plasma. And we applied leptin for 56 days on these animals. Okay, so we started here at day 14 and look at the size of the follicles, right? The triangle here, the open triangles is the low leptin, the low energy with leptin. And the squares are the low energy heifers. So they're pretty close here. As soon as we started the leptin treatment, look at the size of the follicles up here. That's what gave this curvilinear response to the hormonal treatment on the size of the follicles. So it actually, for the first 2 weeks, we had a very strong response to the hormonal application, increasing the size of the follicle. We all got very excited about what was going on, but as you can see, the effect diminished. And by the end of the experiment, the size of the follicles were the same, and there was no effect of leptin on ovulation. This is a survival graph, and it shows that 100% of the animals were in the experiment. As soon as the animal ovulates, it gets out of the experiment, so it disappears from the population. The high energy diet is this line here, and you can see the animals are ovulating and getting away from the experiment. And the other two lines represent the low energy group and the low energy plus leptin, and there was no difference in the ovulation age of these animals. Had no effect on the age of puberty. So that's against the theory that leptin is important for initiation of puberty. We were finally able to measure leptin concentration in the blood. That has been a huge problem in Brazil. There was no one with the serum, and and the antibody to measure leptin, and we couldn't ship the blood to other countries because of foot and mouth disease. So we got stuck without being able to measure leptin concentration in the blood. Finally, one lab got this working, and we measured leptin concentration. We saw that there was an increase at the beginning of the experiment, and then a decrease in the leptin concentration in the blood by the end of the experiment. So that didn't make much sense. And finally, we analyzed the leptin expression in the adipose tissue. And you already seen the increase caused by high energy intake, but you didn't see the decrease, the 2.5-fold decrease in leptin expression in the endogenous leptin when you add the exogenous leptin to the animal. So we think there was some sort of feedback, negative feedback mechanism reducing leptin expression, and that caused the reduction in leptin concentration in the blood, and perhaps that could explain why we didn't see an effect. But the conclusion is it did not support our hypothesis that leptin was the limiting factor for puberty. We know there are several factors for puberty, right? It's not only one, but we thought that with these old heifers that went through periods of subnutrition, that is common in Brazil and here in Australia as well, that might be the case that leptin was the limiting factor, but we could not show that. What it did show was that the administration of leptin decreased the expression of the leptin gene, suggesting the existence of feedback mechanism in cattle. All right, our fourth experiment was about progesterone effect on puberty. So that is another model where we can induce puberty, and we wanted to test the hypothesis whether or not progesterone would change NPY receptor expression and G protein couple receptor signaling in the brain. So we had 90 heifers, prepubertal heifers, same diet. After 4 months on this diet, we selected the 60 heifers for our experiment, and 36 heifers were slaughtered. And we actually combined 3 heifers together to get an RNA pool and had 12 pools of RNA for RNA sequencing. So the 15 heifers for each of the treatments with a progesterone device in these 2 groups, 12 days before slaughter. So we add the progesterone device, and both groups at the bottom did not have the progesterone device. We also tried to manipulate the estrogen concentration in these animals, and we did that by removing the large follicles 4 days before the slaughter. So this first group here, 4 days before slaughter, we aspirated the large follicles, and that would reduce the level of estrogen in the blood. Right? So you have estrogen and progesterone effect on these animals. I'm not going to talk about the estrogen effects. I'm only going to talk to you about the progesterone effect. There was no interaction between the 2 of them, and they would complicate a lot my life here. Because you can see a clear difference between the progesterone group, treated heifers, and non-treated heifers. So that's clear, right? High progesterone, low progesterone. What is going on with the estrogen is more complicated, but we'll get there eventually. So for this experiment, we actually divided the hypothalamus in 3 sections. We want to look specific at the preoptic area of the hypothalamus and the anterior area of the hypothalamus. We didn't do much with the posterior area of the hypothalamus. There were about 13,000 genes in each area that were analyzed. And there was a clear difference between the areas in the hypothalamus. So these are 451 genes that were exclusive to the preoptic area of the hypothalamus. Only detected in the preoptic area of the hypothalamus. When you put that through David to find the pathways that were enriched in this area, There's a nice one, there's called, I cannot read, neuroactive ligand receptor interaction, right? And I'm going to show you some genes that come from these pathways. Neuroactive ligand receptor interaction. So that sounds cool. And look at this guy here. The NPY receptor is actually called, this gene is G-protein coupled receptor 83. And the description in the UniProt is an orphan receptor. We don't know much what it does, but it could be a neuropeptide 1 receptor, right? And that is exclusive to the preoptic area of the hypothalamus. When you look at the gene expression, progesterone had no effect on anterior hypothalamus and had a clear effect on the preoptic area of the hypothalamus, where 34 genes were upregulated and 85 genes were downregulated. When you look at the heat map, you can clearly separate the animals without progesterone from the animals with progesterone in the preoptic area. The genes that were upregulated, when you put in David to look for pathways, nothing really interesting there, just basic, 9 genes, but I couldn't find anything of significance to the reproduction development that we're talking about. But when you look at the downregulated genes, you see the same pathway that I described before that was exclusive to the preoptic area, neuroactive ligand receptor interaction. 5 genes in this pathway, and looking at these genes, first one is HTR1E gene, and it's the similar story as the beta arrestin that I've talked before with the nutrition experiment. It's a gene that modulates the G-protein coupled receptor signaling, right? A similar one is HCR-TR1 gene that was downregulated by progesterone. It also modulates the G-protein coupled receptor signaling. And look at the third one, that little guy there, G-protein coupled receptor 83, that could be a neuropeptide 1 receptor gene that was exclusive to the preoptic area, nowhere else in the hypothalamus, and was downregulated by progesterone, right? So similar effect as in genetics and nutrition, progesterone is decreasing the sensitivity of the preoptic area of the hypothalamus to the bad guys, the guys that inhibit puberty. So as soon as you remove the progesterone device, that's what happens in the field. You add the progesterone device, you block puberty, but as soon as you remove progesterone, then you stimulate the initiation of puberty in these animals. And one reason for that might be that because of the progesterone exposition, the brain would be less sensitive to the negative feedback and therefore would initiate puberty. Okay, so the conclusion for this fourth experiment is that the progesterone is targeting exclusively the preoptic area of the hypothalamus, and it supports the hypothetical model that the sensitivity of GnRH neurons to NPY is important in modulating puberty onset. All right, some take-home points for you. All 3 mechanisms that we've studied modulating age of puberty, genetics, nutrition, and progesterone, alter NPY signaling in the hypothalamus, reduce NPY sensitivity, and therefore lower NPY inhibition of GnRH secretion. Both genetics and nutrition increase leptin expression in the adipose tissue, but contrary to what we wanted, leptin infusion did not induce early puberty. So, in summary, we still believe that high energy intake, well, we know high energy intake induces puberty, we know body fatness is important, but we still don't know, we're back to the quote at the beginning. We are still ignorant of how the brain senses the nutritional status and the metabolic reserves of the animal. What we know is that the sensitivity of the GnRH neurons to the negative effectors, such as NPY, is probably really important, and it can be modulated by genetics, nutrition, and progesterone. All right, I want to thank FAPESP and CNPq for funding the research and the trio of female grad students that did the research. Thank you.